Hi, I'm Caroline Clark, conference producer at Technology Networks, and I'm here to introduce the final talk of the day, network-based strategies to study context-specific human cell signaling. I'm pleased to welcome Evangelia Petsalaki as your presenter. Evangelia completed her PhD on structural bioinformatics, predicting peptide binding on protein structures. During her postdoctoral research, she worked on a diverse array of projects, from Rho signaling, proteomics, phosphoproteomics, to yeast genetics. She is currently a group leader at the EMBL EBI, where her group focuses on the study of the principles underpinning context-specific cell signaling responses in human, mainly using network-based approaches. A warm welcome to you, Evangelia. Following the presentation, we will have a Q&A session, and we will welcome any questions that you may have. Please ask questions using the Q&A system to the right-hand side of the video player. You can also use this feature to connect with attendees and discuss the talk, so feel free to take advantage of the chat system by introducing yourself and sharing your thoughts during Evangelia's presentation. If you experience any technical difficulties during the presentation, please request help using the technical help icon on the left-hand side of the player. Now, without further ado, I will hand over to Evangelia to begin their presentation.
Thank you very much, Caroline. As Caroline mentioned, my group is interested in cell signaling. Just to bring everybody on the same page, I'm going to start by explaining what cell signaling is. So when we talk about cell signaling, we describe the processes that happen in the cell in response to changes in its environment. Typically, we think about processes or pathways that have a part that perceive the signal or the change in the environment, a part that relays the signal, and a part that causes the change in the cell's behavior. Traditionally, we have studied cell signaling pathways very well, because anything that goes wrong with cell signaling pathways can lead to disease, including cancer, diabetes, many other kinds of diseases. So it's very important to understand them both at a molecular level and at the systems level. All the knowledge that we have accumulated for signaling pathways throughout the years has been collated in databases that have been manually curated. And we present them kind of like these cartoons with a kind of a one signal, one response paradigm for simplicity and for being able to interpret our results within the context of the prior knowledge. However, in reality, this is not how signaling actually works. So first of all, there is a lot of crosstalk and feedback loops in between these so-called signaling pathways, and this is not captured in our current view. Secondly, the response can be very different depending on whether there is a specific mutation background, the cell state, the cell type, its environment. And so even with the same signal, if it's a different cell type, you can get completely different responses. And this also, the context information is not captured by our annotated pathways. And finally, there's a very big so-called dark space of signaling. So there's a lot of interactions and processes that happen that we know very little about, because the majority of our knowledge is accumulated based on kinases or proteins that do this kind of signaling that are very well studied. So here you have the number of publications associated with this kind of kinases, and going upward here and here, and you see that the regulatory relationships between these proteins, the most of the ones that we know, are accumulated here in the top right-hand corner. So there's a lot that we don't know about cell signaling, and so when we only base all of our analysis on these kind of annotated pathways, we risk missing out important and new information. So there is a need for data-driven approaches to study cell signaling, and this is what my group does. And we try to use data-driven approaches to study the rules underpinning this kind of context-specific regulation of human cell signaling. This is very important, for example, in medicine or aesthetic biology, because then we can know, for example, why a patient doesn't respond to a drug, whereas another patient of the same disease does, or we can design custom systems in synthetic biology to respond and produce things that the way we want. So it's important to understand these kind of rules. We use different kind of unbiased omics data sets, so phosphoproteomics, CRISPR-based essentiality, other transcriptomics, imaging data, and here we have our major questions that we target in the group. So, for example, if these signaling pathways that are annotated are not the unit of cell signaling, what is? how these units interact with each other and with other regulatory layers of the cell, and how at the end of the day, context-specific signaling results in specific phenotypes. And this is the interest of my group in general. And today I'm going to talk about two projects in the group. So in the first one, we use gene expression data that is paired with phenotype data to identify phenotype-specific networks that we can then use to make discoveries about the specific phenotype. And then in the second project, I'm going to talk about a network approach that we have developed to perform in silico perturbations so that we can identify targets for disease. And in this case, we used endothelial dysfunction as an example. So starting with the first project, this has been published last year. It was work led by Charlie Barker, a PhD student in my group. And the whole idea here is that there's a lot of transcriptomics data sets available that are paired with phenotypes. However, transcriptomics does not equal cell signaling because it's basically the output of signaling processes. And so the question is, can we devise a way that we can get phenotype-specific signaling networks from these transcriptomics data? And this is just an overview of the method, but I will walk you through it. So, excuse me, as a cell study, we used breast cancer cell shape. And why did we choose to do this? Is because cell shape is a very important prognostic marker for breast cancer patients. And has recently become apparent that it's not only signaling in the cell that regulates the cell shape, but also cell shape itself that regulates cell signaling. And so the cell shape determines a lot of things like potential for metastasis and all this kind of information. So it's quite important to understand and potentially modulate signaling that regulates and is regulated by cell shape in breast cancer. And so to study this, we started from two kinds of data. So first of all, we took data from 14 cell lines, breast cancer cell lines. One of them is healthy, and the 13 were breast cancer. And then this was RNAseq data publicly available. And also there was paired cell imaging data, high content imaging data that gave us multimodal phenotype information about length, cell width to length ratio. So you can see here 10 different parameters that we had for the cell shape of these cell lines. So what we did, we said take a bottom-up approach, so not based on the pathways in the first instance. So for that, we used an algorithm called WGCNA, which gives us a co-expression network. So we find which genes are expressed together, and they group them in groups that are co-expressed together. And then we associate these groups to the subphenotypes. And this gives us the so-called modules, which are colored like this. And these are correlated positively or negatively with the different phenotype cell shape variables. So the benefit of doing this kind of bottom-up approach is that we can identify functional units in these clusters that are not covered by the existing pathways. And also we are able to break the multivariate phenotype of cell shape to different subcomponents to give us modules or functional units that regulate specifically these kind of components of the phenotype. To highlight the importance of this bottom-up approach, here's a plot. So these, by default, from the method, you get like some color associated with them. They're not annotated. So we did an enrichment analysis to try to annotate them. And you see that some of them can be annotated with a function, such as, I don't know, ECM organization, insulin signaling, RAP1 signaling, but many of them have very cryptic or no annotation. So like this module not annotated 3, 4, chromosome 7. So you can see that these are modules or units that we would not be able to identify if we just took this original data and did an enrichment analysis, let's say, with existing pathways. So this gives us the opportunity to go, let's say, a little bit into the dark space. So how do we then proceed? So as I said, these are the different genes that are present in the different modules that we identified. And then when you start looking into, okay, what kind of transcription factors are known to regulate these genes, you see that where there was no overlap, suddenly we start having overlap between the modules. And then if you go also above to the pathways, you can start getting a picture emerging of processes that are regulating or relevant for cell shape regulation. And here you see some pathways as a sanity check, where if you look in the literature, all of these are known to previously associated with cell shape regulation. So we were confident that our analysis is able to at least reproduce to some extent to our knowledge. So we have this pipeline and we have these modules, and now we want to place them all together in a unified network. To do that, we took the transcription factors that we identified from this bottom-up approach. We took the pathways in which these transcription factors are included or annotated to participate in. We also performed a differential expression analysis between the different cell shape groups of the cell lines themselves to be inclusive. And we put them all, these gene lists together, and we used an algorithm called price collecting Steiner forest to extract a minimum network that includes as many of these genes as possible, minimizing the edge numbers from a database called Omnipath that includes all signaling pathways, let's say, collated together. And the resulting network comprised about 600 nodes. And here's a word cloud with, let's say, the key nodes, the ones that have the biggest betweens and centrality. And you can see which ones are the major players in this network. Okay, so now we have a network that we believe has the major components that regulate cell shape in breast cancer cells. Is it actually representative of cell shape? Luckily for us, the LINX consortium in the Broad Institute had performed experiments where they had 6 breast cancer cell lines, and they had treated them with a library of small molecule kinase inhibitors and taken this kind of high content imaging measurements. We also had information about the affinity of the different drugs for different kinases, because they didn't only have one target necessarily. So the first check of whether a network indeed is representative of cell shape is the following. So we hypothesize that if it is indeed representative, if we target kinases inside our network, they should impact the cell morphology components more than if you target kinases outside the network. And this inside the network is the blue-green bars, and the outside the network are the red ones. And you can see that for these black subphenotype components, it's always significantly higher perturbation. Whereas for these red ones, actually, you don't have a significant change. And this is very interesting because these guys were not actually included in our training set. We didn't have information about them. So this means that our network not only captures cell shape regulation for these phenotypes for which we did have data, but it's specific to those, and it's just not just generic cell morphology, specific to the input that we gave. And taking this a step further, we checked whether the importance of the kinase inside the network also matters for having impact on the phenotype. And so we devised this score where this score, which basically captures how many of the targets of like a drug are included in the network and what is their centrality, so how important they are. And we found that this score has a moderate correlation that is significant in all the black subphenotypes and not in the red subphenotypes, indicating that again our network indeed is enriched or represents the regulation of cell shape in breast cancer and is specific to the components that we provided. Okay, so now that we're confident that our network is good representation of the components regulating cell shape, we wanted to use it for studying exactly identifying new players for cell shape regulation. So in cancer, we have in breast cancer, there are two kind of major morphologies: the basal-like cells that are kind of disconnected with each other, and the luminal-like cells. And the one of them is more has a worse prognosis for patients than the other. So what we did is we took these kind of cells, we identified transcription factors that are found to be differentially active in the one group versus the other. We mapped them onto our cell shape network, and we used something called network propagation to identify network regions that are affected by these phenotypes. So basically the network propagation principle is that if this node in the network is affected, probably its neighbors are also going to be affected, or this node will affect the neighbor. So with this principle, you end up putting on your seed nodes on the network, upregulated, downregulated, and then you diffuse, let's say, this effect in the network, and you identify signatures of potentially important nodes for the phenotype. And here we have the results for the basal-like propagation and the luminal-like propagation. And you can see in the both of them, we have this gene expression module that we had annotated as RAP1 signaling module. That is the top hit for both of them. And we also have additional genes that are actually, a lot of them known indeed to be important for cell shape regulation. So what is this RAP1 signaling module? Because we annotated it as such, but as I mentioned, our modules do not correspond one-to-one to actual signaling pathways. So it includes, of course, RAP1 components of the RAP1 signaling pathway. That's why it was enriched in this, and we annotated it as such. But it also includes components of the RAS signaling pathway, HRAGE, HIF1 alpha, and some other genes that is good to know what is in there. So I'm showing this to really show you that this is a functional unit that is not really corresponding to an unrelated pathway. And we said, okay, we found this. Is it actually important? Is it actually relevant for cell shape regulation beyond these 13 cell lines that we identified? And to do that, we correlated the activity of the transcription factors that we know are relevant to cell shape. And here you see a few of them with the RAP1 expression across 86 cell lines this time, for which we didn't have imaging data, but we had RNA-seq data. And you can see they are all positively correlated with all of these transcription factors. And this guy is a transcription factor that negatively regulates NF-kappa B. And so that's why it has a negative correlation. And if you take it to the patients, again, here we see that we have a highly significant correlation with transcription factors, again, that are known to be regulating cell shape. And some examples, again, are NF-kappa B, you can see here, TID1, you can see all of the guys that we have here and a few more. So we were very pleased to see that they seems to be also relevant in patients. And indeed, if you look at the morphology that is annotated by pathologists for breast cancer patients, we see that in this morphology that is called metaplastic carcinoma, which is the worst in terms of prognosis for patients, this RAP1 gene expression module seems to have the highest expression. So so we are convinced that there is something to look into in this RAP1 signaling module, and placing it into this kind of mechanotransduction pathways. RAP1, the protein itself, is known to be linked to mechanosensitive pathways, and it is also known to regulate NF-kappa B, but is not known how in different contexts than in mechanotransduction. NF-kappa B is a major regulator of cell shape, so we believe that the module that we have identified serves to link this kind of mechanosensitive pathways with the NF-kappa B in the regulation of cell shape. And here I have annotated some other genes that we also found to be important, and many of them are indeed known to be relevant in this process, suggesting that our method was able to capture actually known biology about the cell shape and also provide new insight. And just to give you an idea how potentially we could identify the links between this RAP1 module and NF-kappa B, here you see components of hippo signaling, Wnt signaling, and this could be playing a role in linking this kind of RAP1 module with NF-kappa B. So we developed this method, we applied it in cell shape, and now we're wondering, is it applicable to other systems? And so for that, Ioannis Kamzolis in my group, he was working on non-alcoholic fatty liver disease. We wanted to see if we can apply it to a disease phenotype rather than a cell shape, which is much, much more complex. To bring everybody on the same page, non-alcoholic fatty liver disease is a disease that starts with accumulation of fat in the liver. About a third of the population in the Western world is believed to have this kind of early stage NAFLD, and then a fraction of them will progress to steatohepatitis. Steatohepatitis, and even smaller fraction will progress to cirrhosis or hepatocellular carcinoma, and eventually will need transplant or may also die. And this is a disease that is increasing in prevalence, and a lot of the transplants for liver are coming actually from patients of NAFLD. So there's an increasing need to understand how the disease progresses and what are the can we target it, basically. And also we need biomarkers to identify which ones of these patients will proceed to the next stages. And so to study this kind of mechanism of disease progression, we took a slightly different approach than most people. So most people treat it as 3 to 7 discrete stages, depending on the paper that you look at. And this is subject to the pathologist's judgment based on the histology annotations. However, the disease is not discrete, and it falls rather on a continuum and perhaps even multiple continuums. So we hypothesize that if we use kind of a patient trajectory approach, then we would be able to get better information about how the disease progresses. And so we used pseudotemporal ordering of the patients in order to place them along the continuum of the disease progression. And these are the previous annotations for their disease stages. And you see that the mean of the NAFLD stage along the trajectory actually follows this kind of progression from green to light yellow greenish, which indicates, here we have control or early stages, and the disease progresses as you go to the left. So with this in hand and our previous method in hand, we said, okay, can we walk along the trajectory and using kind of a sliding window approach to make kind of a video of the networks and how they change along the trajectory. So we again, this time took the RNAseq from the patients and the histopathology score for the different subcomponents of the disease. We ran WGCNA. Instead of correlating it this time, we used the linear model to associate it with different subphenotypes. And then we ended up with kind of a little network here of modules that are associated with the disease. So red is positive correlation, association, and green is negative. And then if we look at, if we do an enrichment of these pathways, of these modules as they are associated with the disease, we see here that it recapitulates what we know about the histology. So for example, the status starts somewhere here and it starts getting worse. And then the ballooning starts next. And then is the fibrosis with a NAS score kind of providing a summary of all of this. And it kind of agrees with what we know from the histopathology. Sorry, it takes a bit of time to load the next slide. There it is. So then, okay, we starting again from the transcription factors, we went along the trajectory using sliding windows, and we calculated a score that represents the transcription factor activity times the module enrichment in the specific sliding window. And here are the different transcriptional factors that we found to be enriched and significantly associated. And you see how they change. So we have this block that is constantly on, this block that starts low, but very fast goes up, this block that goes down. And here are known associations with NAFLD. And you can see that many of the transcription factors that we found are indeed known to be relevant. Here we have a little network of these transcription factors, and each of these edges indicates that they are functionally similar to each other. And so you see that we have here a little cluster of very functionally similar transcription factors and other ones that are a bit more outliers. This is, let's say, their score in sliding window 1, and by the end you have many more of these transcription factors that are either upregulated or downregulated. But as I said, we wanted to go upstream to the signaling, and to do that we're using a algorithm called MaxFlow. So for each window, we're taking the receptors that are differentially expressed, the transcription factors that we find to be active, and we try to identify the paths on our NAFLD network that are most likely to be used between these. And we collate these all in a table, and we assemble networks per sliding window. And it's kind of what it looks like. This is the 95th percentile of the edges. Apologies, missing here the end of the word edges. And you see here that you start in sliding window with a small little network. And anything blue is the new things that are getting added. And you can see in sliding window 5, we have some addition of a little module here. And in sliding window 10, we have the most addition of nodes. And from 20, we have a few more. And going forward, we have addition of very few nodes and some small fluctuations of nodes coming and going. So this is very interesting. It indicates that a lot of the changes that happen in the disease progression happen rather early on. And we can now go in and dig in and look at the individual genes and see which ones might be important or relevant to target. For example, this DKK1 arrives and links a whole new set of genes with this kind of module with the previous network that was there. And we know it's a Wnt signaling agonist, and it has a role in inflammation. And it is a marker for hepatocellular carcinoma. So it's something perhaps to look into. So this is kind of the way we generate these networks. And if we cluster the transcription factors according to their behavior, so these guys go up, down, then up again. These guys are kind of constantly mildly up. These go up in the end, etc. We can identify signaling pathways or processes associated specifically with these clusters and study them individually. And now we are in the stage where we are trying to identify more hypotheses that we can test from this data. And specifically, we want to associate these changes in network change with changes in the cell type composition of the liver, because we know that there's a lot of inflammation happening, so a lot of changes in the cell type composition. And we want to know which ones of the changes we see in the network are due to just change in cell types, or they are actual deregulation that we can actually target. And so to summarize this part, I showed you a method that we have developed to extract phenotype-specific networks from transcriptomics and phenotype data. This method uncovers relevant, context-specific, or phenotype-specific functional units that go beyond our current annotated pathways. And then we can identify subcomponents of multivariate phenotype. We are in the process of doing further development of the method to provide even more robust modules. And I showed you that we have used these kind of networks for network propagation or max flow, but you can also be used, for example, as prior knowledge network for modeling, since they are directed and signed, or for any other network analysis application one wants. I showed you two applications. One was in breast cancer cell shape, where we identified the breast cancer cell shape specific network and identified a module that was enriched in RAP1 signaling regulators as central to the phenotype. And I showed you an ongoing application on non-alcoholic fatty liver disease, where we created a disease trajectory based on the patient data, and we're using our method to identify the mechanism of the disease progression. And so this is the first part. And so in the second part, I want to present to you another approach we took to again integrate different modalities of omics data into one context-specific network, and and then I tried to create an in silico perturbation approach to identify targets. And this was work led by Igor de Souza in my group, and with contributions from Girolamo Giudice. So then this endothelium is a very special kind of tissue in the body, because it is everywhere, and so it's subject to a variety of condition, a variety of pressure, of shear pressure, of different contexts throughout the cell, and so it has to deal with all of these, and it has many important functions. So other than being a barrier between making a barrier between the blood and the different organs, it also regulates many different processes, and so it's very important that it functions correctly. So when it doesn't function correctly, it basically results in disease, and in fact it's a hallmark of many, or if not most, of the cardiovascular disorders. And so we wanted to study the endothelial dysfunction and identify what are the networks or the molecular processes underpinning this, and see if we can target it directly rather than targeting symptoms of different cardiovascular disorders rather than the actual cause. And to do that, we took an integration approach. So we used public data sets from human aortic endothelial cells that were exposed to different conditions simulating and causing endothelial dysfunction. And this was ATACseq, ChIP-seq, and RNAseq data. And we started from that. We first analyzed the ATAC and ChIP-seq data, and we took the overlap of the peaks, and which were about 6,600. We identified sequence logos that were enriched, and then found 29 transcription factors that were predicted to bind to these motifs. Taking this into account, along with differential expression analysis from RNAseq, we took the transcription factors identified here and those genes from the RNAseq that are known to be regulated by these transcription factors, and we assembled a small gene regulatory network of 86 genes. And then, using network propagation on a protein interaction network, we extracted a larger network, I think about, again, 500, 600 nodes that we believe represented endothelial dysfunction. It's very difficult to validate to be 100% sure that this indeed is an endothelial dysfunction network, but as an indication of this, we took single cell data from patients from normal versus atherosclerotic plaque endothelial cells, and we found that there was a significant enrichment in our network of those genes found to be deregulated in these conditions, at least supporting that indeed this is enriched on genes relevant for endothelial dysfunction. So now that we had this network, we wanted to do our in silico perturbation. So what we did is for those 86 endothelial dysfunction genes, we removed each one as if simulating an in silico knockout and regenerated the network. And then we compared then the resulting networks with the original one using the Jensen-Shannon distance. And by combining this score with random walk score and the between centrality, we were able to get a ranking of our genes. And so from among the top ones, we selected 17 for which there was a drug available, and we performed in vitro tests. And here, the ones that are, let's say, transparent did not have a significant effect, but we identified about 7 of them that had an effect in at least one concentration of the drug in at least one of the conditions that we tested. This one represents oxidative stress, and this one is inflammation. And eglin, the molidustat, which targets eglin-3, actually show the phenotype in both the conditions. And here are the respective images, different concentration of the drugs, and this simulating condition of endothelial dysfunction and the control. And you see that with the drug, we see an effect in both the conditions. We're now following this up with shRNA measurements, but this work is already available in bioRxiv if anybody's interested. And to summarize this part, I showed that we combined different omics data sets to identify a set of endothelial dysfunction genes, which we use then to create an endothelial dysfunction network. We developed an in silico perturbation approach, which identified 17 candidates for drug repurposing. And of course, there were also other genes, perhaps that were even higher ranked that there was no drug available, and we are following those up with shRNA. And we found that 7 of the 17 drugs improved at least one of the 2 phenotypes tested, and the one improved both of the phenotypes tested. And with this, I would like to finish, and I just want to summarize the general message of the talk. So cell signaling happens through complex and context-specific networks, and our current way of studying them using the annotated pathways as the gold standard does not allow us to study this kind of context-specific regulation. There's a lot of valuable transcriptomics data sets there that can be used to study cell signaling, but we cannot immediately translate them to signaling networks. But by anchoring our analysis on transcription factor activities, we can use them to extract context-specific signaling networks. And in my group, we're using different data-driven approaches to do this. And I showed you 2 examples. Using 2 examples. In the first one, we identified a cell-shaped network for breast cancer, and we found RAP1 module to be an important cell-shape regulator. Then we also identified an endothelial dysfunction network, and we're able to identify critical nodes that have potential for drug repurposing. And with this, I would like to finish and thank current and past members of my group, and Caroline and all the organizers for inviting me, and you for your attention.
Well, thank you very much, Evangelia. That was fantastic. You've got some great research going on there. We are likely going to have lots of questions from the audience, and I can see that we've received some questions already. So if you don't mind, we'll move straight on to the Q&A now. So your first question is, what are the biggest challenges you face when you generate context-specific networks?
Thank you. So the biggest challenge that we face is that there's no possibility, or there's very limited possibility for benchmarking them so that we know for sure if they are correct. And this is a major limitation. So we always have to be creative with this. And also, for some of the methods, for example, for the method I showed you, the first one, depending on the data sets that you have, you might already rely on the existing pathways, for example, to extract the nodes relevant to the transcription factors, and this reintroduces some of the bias that is, again, not context-specific information there. So if we have multi-omics data sets that we can use to integrate the different layers, then this improves the situation and gives us more accurate context-specific networks. But yes, these are the 2 major limitations.
That's great. Thank you for that. And moving on to your next question. How generally applicable are these methods?
Yeah, so I think it all depends on what ability you have for validating the resulting network, because you should always have a sanity check for this. But in general, they are very widely applicable. So the first method that I presented, as I showed you 2 case studies, it can be applied to any method that has paired omics, any omics, not necessarily transcriptomics, paired with any multimodal phenotype data. And the second method can be applied again for the integration is straightforward. So we basically just got a consensus list for the seeds and extracted the network with network propagation. And the in silico method can also be applied to any network of interest. But one thing to keep in mind is that the in silico perturbation gives us information about how important the node is in the network and not necessarily the disease. So it's always just a prioritization exercise, which has to be then validated in the lab.
That's great. Thank you for that. I think we've got time for a couple of more questions. And your next question is, are these methods available for people to use?
Yes, both these methods are all the code is available. The first method is published in Genome Research, and the code is available in Zenodo, and also the other method the same. They're publicly available with the preprint. We also are happy we have new developments for the method. So if anybody's interested in using them, feel free to reach out and we can help. We can give you the latest version, let's say, even before it's published. Yeah.
That's great. Thank you very much. And we've got time for one more question. And it is, can you integrate additional data modalities in these approaches?
Yes, that's a very good question. So the first, especially with the first method, it's extremely flexible because you can, for example, here we derive the transcription factors from the transcriptomics data. But if you have more specific information, for example, ATACseq or ChIP-seq data, you can put that there instead. Then we got the pathways from just the databases. If you have phosphoproteomics datasets or proteomics datasets, you can use that, and then your result is going to be much more specific to your conditions. For the second dataset as well, you can integrate whatever information or data modalities that you like to extract your initial seed nodes that you can proceed. Yeah.
That's great. Thank you very much, Evangelia. Unfortunately, that's all the time we have for today. Any questions that we didn't have time to get to will be sent to the speaker after the event. And as with all the talks in this event, the talk will now be made available to view whenever you want for the next 30 days. So that's it for the Innovations in Disease Modelling Online Symposium. As mentioned throughout the day, all the talks from today's event will be made available to view on demand for the next 30 days. You can access these talks via the same link you used to access them today, and we will be sure to pass on any further questions you submit through the system to the relevant speaker. So that just leaves me to say thanks again, and we will see you again at our next online symposium, Cell and Gene Therapy 2023. Goodbye.